Hi, I'm Eliska, and in this presentation, I'll talk about our tool called ENNGENE that can help you build neural networks for diverse classification problems on genomic data. This tool has been developed in our RBP Bioinformatics lab as part of Central European Institute of Technology in Brno, Czech Republic. So the basic idea behind the tool is creating neural networks without the use of programming, so purely using the graphical user interface. In this chart, you can see the simplified data flow of the application. The application is divided into multiple modules. In the first module, the input coordinates from the BAT files are prepared so that they can be given to the neural network. In the second module, the architecture and other parameters are set up, and the model is trained with the pre-processed data. In the last modules, the trained model can be used either to classify novel unseen sequences, or it can be evaluated on different data sets. So in the first module, the given coordinates are mapped to the provided reference genome file. From the original input sequence, input coordinates, we can obtain up to 3 different input types. So the first one is a pure DNA or RNA sequence that is later on one-hot encoded, or the mapped sequence can be folded, or the secondary structure can be predicted by the Vienna RNA2 package, and this is also later on one-hot encoded. The last option is that the input coordinates are mapped to the provided conservation phyloP files, and this way we obtain the evolutionary conservation score. In the second module, the network architecture can be customized. The user can choose from convolutional, recurrent, or fully connected layers. Also, each layer can be customized, as well as other training parameters such as batch size, optimizer, or learning rate, for example. Here you can see how the different input types that I've already mentioned map to up to 3 different branches in the model. So each branch consists of arbitrary number of convolutional layers, and these branches are then followed by connected part of the network that can be composed either of recurrent or fully connected dense layers. During the training, you can monitor the accuracy and categorical cross-entropy loss in an interactive plot directly in the application. These metrics are also exported after the training is finished. Also, 2 most commonly used evaluation metrics are exported. Those are the receiver operating characteristic curve and precision recall curve, both adjusted for the multi-class classification problem. Also, TensorBoard files can be exported. The TensorBoard is an external tool that can help you visualize or compare metrics throughout multiple runs, or, for example, interactively browse the model architecture. In the last modules, the trained model can be used either to classify novel sequences, or it can be evaluated on different data. To help understand and interpret the predictions, we use the integrated gradients method. The integrated gradients are currently widely used method for interpreting deep neural networks. In our case, it helps us to visualize each nucleotide and to see if it had low or high attribution to the prediction. It's adjusted for all the combinations of all 3 branches so that you can see what input contributed the most. To showcase the ENNGENE application, we chose the RBP24 dataset. This dataset comprises of 21 RNA binding protein target sites from 24 ClipSeq-based experiments. This dataset have been used already by 6 different publications, so it's good to benchmark our model's performance. The deep learning methods in general have been applied on the RNA binding protein target site classification problem since 2015, with the DeepBind project as the first one using convolutional neural networks. Since then, many different architectures have been tried, with the most common being the combination of convolutional and recurrent layers. Here we hypothesized that given the diverse nature of RNA binding protein binding specificities, it would be optimal to specify the architecture for each protein separately. We can say that we have succeeded, as we've reached the state-of-the-art performance for all of the proteins while improving the predictive performance on more than half of them. We also significantly improved the average AUC-ROC value for the RBP24 dataset. Said that, we also had some observation that were common for all the proteins across the RBP24 dataset. For example, the predicted secondary structure did not improve our model's performance. So we could say that it was redundant to the sequence itself. On the other hand, the evolutionary conservation score improved our performance in almost all of the cases. Also, even though the convolutional and recurrent combination of the convolutional and recurrent layers is the most often used combination so far, in our case, using the recurrent layers did not improve our performance compared to using just the fully connected layers. Said that the logical continuation of this would be to train one common model for all the RBPs together, and then use the transfer learning and fine-tune the model for each protein separately. So that's all from me. I hope the presentation was clear, and if you're interested in the ENNGene tool, I'll be happy if you contacted me, or you can check our GitHub page where you can download and use the tool or read the documentation.